Hey guys, welcome back to Tuesday, April 21st. Today we're just going to review the stuff we did yesterday, talk a little bit about DNA replication. So what did we talk about yesterday? We talked about the structure of DNA, right? And we started with the function. So did we remember what the function of DNA is? It has to be able to replicate. It has to be able to store information. It has to be able to code for the production of proteins, and it has to be able to mutate. Not all mutations are bad. That mutation allows for variety, it allows for evolution. It allows for organisms to adapt to a changing environment. Right? So that's the function of DNA. The shape of it, it had that sort of double-stranded twisted ladder action, also known as an alpha helix, right? And that those little DNA strands, those make up genes. Well, they don't really make up genes. Let me get rid of that. Genes are segments of DNA. Much better way of saying it. I don't want to steer you in the wrong direction. And then we had the bases, right? Oh yeah, also the nucleotide. We need to know the structure and what that's made up of. I think we covered that before. That's a deoxyribose, right? So it's missing one oxygen. That's the sugar, which is called deoxyribose. And remember, back when we were in school, we did sugars, carbohydrates. Ribose, OSC is a sugar. Over here, we have a phosphate. And over here, we have our nitrogenous base. It's called nitrogenous base because it has a nitrogen in it. The bases, you have A, T, C, and G. Adenine, thymine, cytosine, guanine. A pairs with T, T pairs with A, C pairs with G, G pairs with C. Now, let us talk about DNA replication. DNA has to replicate. If DNA doesn't replicate, then we no longer exist. We have to store this information. You hear that bird up there? That bird, it has all of its DNA. It has all of its DNA. All of its DNA is able to replicate, and then it can heal itself. It can make more feathers. It can make, I guess, more, I don't know, tongue if it has cells, so it can go beep, 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 which drives me nuts as I'm making this video. But the way it works, there's a process. So let's say we have a nice strand of DNA, and I'm going to use black for my nitrogenous, for my sugar backbone. I'm going to use blue for that base, and we'll just use blue all around. Doesn't matter. Blue all around. Okay, so in order for DNA to replicate, we have to go through a few steps. The first thing is it has to unwind. So step one is it unwinds. So it goes from that little wrapped up double helix to that, to what you see right there. Oh, down. I don't know where my face is going to be at this point, but it'll be somewhere. After that, it is going to unzip. So let's say I have A, T, C, G, A, T. Okay? It's going to unzip. Those hydrogen bonds that are holding it together are going to come apart. Going to come apart. So then I'm going to have over here my see this? You still hear that bird, right? That bird is still going. We got this red, a cardinal. We call it red. It always hangs out on my fence over there. It's weird because I'm pointing the other way, but that's the direction that's in. Anyway, so I have my A, my C, my A, and I have my T and my G and my T. So they are not connected anymore. They're unzipped. So there's this enzyme. You know how I like to draw my enzymes, right? The enzyme is called DNA polymerase. Remember, ASE is an enzyme. DNA polymerase comes in and actually adds bases. So it will add bases over there. I'll just draw it over here just because it's easier. It'll add bases over there. And so what you're going to end up with... Bug just went on me. The pleasures of being outside. Drawing over here, filling the silence that is my backyard. There we go. So now we have ACA, TGT, ACA, TGT. That DNA polymerase goes and puts the bases on. So step 3, DNA polymerase adds the complementary. That just means the other base, the base that matches it. So if you had A, it's going to add the complement, which is T. So it's going to add the complementary bases. But we're not done. We are then going to add an enzyme called ligase. The way I remember it is the ligase links it together. And so an enzyme is going to glue it together, and it zips back up, and then it coils back up. Now, we don't need to know the enzyme. We're just going to put enzyme, zips it back up, and then the DNA coils back up. And now we have 2 separate strands of DNA. So that's all that's going on here with DNA replication. You have 1 strand of DNA. If it looks like this, right, it breaks off. The complements are added. Then you have 2 new strands, and then they just zip up and they wind themselves back. That's DNA replication, and that happens in your cell. When it happens outside your cell, it's a piece of DNA technology called PCR, polymerase chain reaction. It uses DNA polymerase to do this outside the cell. And they can do this in murder investigations, in all sorts of criminal investigations. They use this to, if they get a full DNA sequence of the dinosaurs, they can use this to make more of it. And then maybe you could have your own pet dinosaur, which is not really going to happen. All right, so all I covered today was a review of what we did yesterday, the function of DNA, shape of the DNA, the bases in DNA. And then I just went into a little bit in DNA replication because I know there's a question that was on your sheet yesterday that I told you not to do. Now you can answer it today. Now, why would DNA replication be important? To be able to make more of it. Why would you want to make more? Well, if you want to grow, you need to make more. If you want to solve a case like a crime, if you only have a little DNA, you can make more of that DNA, then you could store it and you have it for future use. So if you have like a missing persons or a murder investigation that you have, and there's some DNA left there, and you're waiting to test it against a bunch of suspects, you don't want to waste it all, but you can use PCR or DNA replication to make more of it. All right, so DNA replication is important to make more DNA, to be able to grow, to be able to heal. And then, of course, outside the cell, PCR can be used in criminal cases. All right? That's all I got for you today, and I will see you tomorrow. Peace!